Now let us see the general histology of muscles. Muscles include skeletal muscle, cardiac muscle and smooth muscle. First let us see the skeletal muscle. This is how to draw the skeletal muscle. It consists of unbranched, long cylindrical fibers, multiple peripherally flattened nuclei, dark and light cross striations are seen. Each cell is long, cylindrical, without branching, and fibers run parallel to each other. Skeletal muscles have transverse striations which are seen as light I band and dark A bands. Dark A bands with light I bands. Hence they are called striated muscles. Striations are not easily seen under magnification. Center of I band is called Z line. Center of A band is light head zone. Dark M line is seen in the center of head zone. Let us see it through a diagram. Dark and light bands, center of I band. This is the I band, light band, and this is the dark band, A band. Center of I band is called Z line. See, center of I band is called Z line, and center of dark A band is called head zone, and the dark M line is seen in the center of head zone. Light band, I band, dark A band, center is known as head zone, and the center of I band is Z line, and the M line is seen in the head zone. The part of myofibril between two adjacent Z lines is called sarcomere, which is the functional unit of muscle between two Z lines is called sarcomere. And A band remains constant during muscle contraction. I and H bands become short. The skeletal muscle is covered by sarcolemma and is multinucleated with flattened peripherally positioned nuclei. Skeletal muscles are voluntary muscles innervated by somatic motor nerves. Connective tissue covering of each muscle fiber is endomysium. This is endomysium. The sarcolemma is also known as endomysium. Bundles of each fiber, such fibers, fascicles, are covered by perimysium. Entire muscle covered by epimysium. This is the entire muscle and it is covered by epimysium, and this is the fascicle. Fascicle is covered by perimysium, and each muscle fiber is covered by sarcolemma or endomysium. These are the identification points of skeletal muscle. Now let us see how to draw skeletal muscle. We need to draw cylindrical fibers like this. It includes like this. We need to draw cylindrical parallel fibers like this, and we need to place flattened nuclei on these lines, and we need to cover with eosin pencil, and we need to trace some lines which are known as dark lines, dark band. So this is skeletal fiber. Labeling include this is known as sarcolemma or endomysium, peripherally flat nuclei, dark and light bands, and this is muscle fiber. Now let us see cardiac muscle. This is the cardiac muscle with short cylindrical fibers with central or oval nuclei, branching fibers seen, intercalated discs are seen. Cardiac muscle fibers are small, cylindrical compared to skeletal muscle fibers. Nuclei are central, single, oval. Muscle fibers are branched. Intercalated discs is one of the unique feature of cardiac muscle. Is the junction between adjacent cardiac muscle cells. adjacent, these are the two cardiac muscle cells, and this is the intercalated disc. It is zigzag in appearance and made up of three types of cell junctions: desmosomes or adhering junctions, gap junctions, and tight junctions. Gap junctions allows electrical impulse to pass from one cell to another and makes the cardiac muscle fiber a functional syncytium. Cardiac muscle is also striated. See, we can see the dark and light bands, and it is involuntary, seen only in heart. It is highly vascular and innervated by autonomic nervous system. These are the identification points of cardiac muscle. Now let us see how to draw cardiac muscle, and we need to draw the cylindrical fibers, and we need to draw branches like this, and we need to draw dark and light bands with eosin, and we need to draw intercalated discs which we need to draw darkly with eosin pencil, and we need to draw dark and light bands and centrally placed nucleus. Now let us see the smooth epithelium, smooth muscle. It consists of flat elongated nucleus and spindle shaped cells. This looks like this. It is non-striated and involuntary. These are the spindle or fusiform shaped cells with central oval elongated nucleus. Sarcoplasm contains actin and myosin filaments without an orderly arrangement. In skeletal and cardiac muscles, the filaments, actin and myosin filaments, are orderly arranged. That's why we can see the dark and light bands, but here we cannot see them. Hence only longitudinal striations are seen, found in walls of digestive tracts, blood vessels, and uterus, innervated by parasympathetic and sympathetic nerves. These are the identification points of smooth muscle. So these are some of the important muscles. Thank you.